Just started. Yeah. Are you not doing biology at all?
I don't be join.
Just agenda.
Good morning. Can you hear me? Good morning. Can you hear me?
Good morning, sir.
Good morning, sir. Okay, so all right, today please mute yourself. Today we are going to talk about, we already spoke about okay, any questions from the last two classes? Any questions? Okay, so we have already spoken about hello? Okay, so we already spoke about organelles, spoke about the cell. We looked at organelles that are not membrane bound, typically the ribosome. And also we looked at organelles that are membrane bound. Then we also looked at organelles that have double membrane, where we looked at the nucleus, mitochondrion, and chloroplast. Mitochondrion is singular, mitochondria is plural. So today we are going to look at mitochondrial and chloroplast from a different point of view. But first, please, if you are not, if you want to talk, hello, if you have something to say, raise up your hand, I will call you. Otherwise, mute yourself, please. Neka Oyechi.
Yes, sir.
Neka.
Please, yes, sir. Good morning. Good morning.
Morning.
Yes, sir. Please, I wanted to ask. You said we are continuing. Sorry, I'm asking the question. The last place we stopped was nucleus. Nucleus. So I wanted to know. You said we are looking at this mitochondria in different direction because we didn't have this mitochondria in the last class. I wanted to know if we are continuing from nucleus.
Okay, but we did finish the nucleus when we were talking about the nucleus.
We just stopped at structure. It is surrounded by a nuclear envelope. That is where it stopped. Then you were now asking class and questions till the class ended.
Okay, so that means that I sent you the slides, right?
Oh yes, sir.
Okay, so I didn't complete the discussions on the slide is what you are saying, right?
Yes, I think so because now we are moving to mitochondria. So I was quite confused. I wanted to know if I should just
Okay.
continue. Yeah.
No. All right. I'll pull up the other slide. Yeah. And then yes, I'll just stop this a minute and bring the other slide. That's D, right? Yes. Sugar. That's my error. I'm sorry. Thank you, Nika. Okay, in the meantime, any questions?
Good morning, sir.
Good morning. Is that Temidayo Daniel?
Yes, sir.
Is that Temidayo Daniel?
Can we get the YouTube link for those that are not in YouTube, sir?
I thought I saw something on that YouTube thing.
No, you've not sent it to the group, sir.
Not the same one. Okay, let me just. Sorry for this. All right. Okay. I hope you have gotten it down.
Ma? No, sure. Also, one bedroom already.
Please mute yourself.
Okay. Is everybody okay now?
Yes, sir.
Right.
Yes,
and the
sir.
YouTubers, they have their thinking. All right. So pardon me. We'll start from the nucleus. All right. So the nucleus is a double membrane organelle present in all eukaryotic cells. All eukaryotic cells have the nucleus. Okay. Double membrane organelle. And that membrane is called nuclear envelope. Nuclear envelope. Okay. The largest organelle and its control center of cellular activity, and it stores this cell's DNA. Like we know, DNA has our genes are stored on chromosomes. Okay, they are in the DNA. The envelope pinched off, the double membrane is pinched off, and where it is pinched off, that place is called the nuclear pore. But it's really a nuclear pore complex because it's a complicated opening. And that's what allows things inside and out of the cell. Okay. Okay, within the nucleus, you have the chromosome that has the genes on it. Okay, and yeah, and this is okay. Here is an electron microscope of the nucleus. Electron microscope of the nucleus. So this nucleus, yes, I hope you can see my cursor. Yeah. Okay. So you have one membrane there, another membrane there, and it's pinched off here. So this gap here is the nuclear pore complex. So this electron microscope shows greater details than ordinary microscope. Okay? So what you have here is ultrastructural details of the nucleus. So going into the nucleus, yeah, you find heterochromatin and euchromatin. Chromatin is the DNA complex with protein. That's all called chromatin. And when this chromatin condenses, it forms the chromosome. So inside the nucleus, the region called the nucleolus. A region called the nucleolus. The nucleolus doesn't have a membrane around it. The nucleolus is just a group of translational active chromosomes come together and form the nucleolus. But the nucleolus also has a nuclear organizing feature. Yes, question.
Yes, sir. I can't hear you. I can't hear you very well, sir.
Okay, how about now?
Yes,
Yes,
sir.
sir.
We couldn't hear you. Your voice was breaking. I think it's the network.
Okay, so I should start from where? You didn't hear me at all.
Yes, it
Yeah,
was
it was
breaking.
No,
quite
just
it was
continue,
breaking.
sir. It's
Okay,
clearer now. It's
I'll
clearer
hold
now.
up the thank you for punishing me. I'll hold up the microphone like this.
Okay, sir.
Okay. So the nucleus is a double membrane organelle and it is found in all eukaryotic organisms. Please note, in prokaryotic organism, it is called the nucleoid. It is not a true nucleus. Okay. Now, this nucleus has two membranes, two lipid bilayers that surrounds it. And those two lipid bilayers are pinched off at the where they are pinched off. That means they join together, they fuse together somewhere. And where they fuse, they leave a gap. That gap is called the nuclear pore. And it's called actually the nuclear pore complex. All right. Now, this nuclear pore complex is charged with responsibility of allowing things in and out of the nucleus. Now, the space between the two membranes that make up the nuclear envelope is continuous with the lumen of the rough endoplasmic reticulum. The space between the two membranes that make up the nuclear envelope is continuous with the lumen of the rough endoplasmic reticulum. So then when you go into the nucleus, what you are seeing is the ultrastructure of the nucleus. This is generated by electron microscope, generated by an electron microscope. So this is the heterochromatin, which is condensed chromatin, okay? And this is the euchromatin. Chromatin is DNA mixed with proteins, DNA complex with proteins. These chromatins will now further roll themselves up to form what we call the chromosomes, all right? So in the nucleus, aside from the euchromatin and heterochromatin, there's now a region within the nucleus that we call the nucleolus. The nucleolus is not membrane bound, right? It is not membrane bound. It's just a region within the nucleus that has transcriptionally active chromosomes that gather themselves together. And they are controlled by the nuclear organizing center. So when newly transcribed DNA, newly transcribed RNA, appear fiber-like under the electron microscope, okay? And so they are called fibrosa. And then when they are complex with proteins, they are called granulosa. So within the nucleolus, okay, you have the nuclear organizing region, you have the fibrosa, which is fiber-like nucleic acid. Then you have the granulosa, which is looking like granules because it has been complex with proteins. Now, this nucleolus is where the subunits of ribosomes are made and assembled. They are made and assembled, and then they are shipped out of the nucleus, and then they are coupled together finally outside. So these building blocks of ribosomes are made here in the nucleus. Okay. All right. So then underneath the nuclear envelope, you have what gives structure, what gives shape to the nucleus, and that is the nuclear lamina. The nuclear lamina. The nuclear lamina. The B-type lamina is closest to the membrane, while the A-type lamina is closest to the nucleoplasm. Everything bounded by the nuclear envelope is called nucleoplasm. So the nucleus stores genes on chromosomes, organizes genes into chromosomes to allow cell division, transports regulatory factors and gene products through the nuclear pore, and produces RNA, which is the messages. Because DNA is double-stranded, RNA is single-stranded. DNA is not allowed to leave the nucleus. So instead, DNA encodes a mRNA, and that mRNA can go through the nuclear pore complex. And okay, I've said it produces ribosomes, but the ribosomes here are the small subunits, the small parts that make up ribosomes. They are produced there, and then they are shipped out of the nucleus, where they assemble themselves together to form the large subunit and the small subunits. And they organize uncoiling of DNA to replicate genes. So you have the heterochromatin. Heterochromatin is transcriptionally inactive. Heterochromatin is transcriptionally inactive. Euchromatin is transcriptionally active. And here you can see the nuclear pore, where the two, this is a one membrane, this is another membrane, and they pinch off. This is another membrane, and it pinches off. And you have the nuclear pore. You need to mute yourself on entry. Chidera, your hand is up.
Sir, please, can you slow down, please? I think we are
okay.
You are very fast. Please,
all right,
sir.
all right. Okay, so all right then. Functions of the NECA. You raised your hand.
Okay, sir. Sorry, I wanted you to recap, but then you went back, so don't worry. Thank you.
Okay. All right. Function of the nucleus. The nucleus stores genes on chromosomes. The nucleus organizes genes into chromosomes to allow for cell division. The nucleus transports regulatory factors and gene products via the pores. Okay, so what are gene products? I'm sure Dr. Fakoide has taught you the central dogma. Yes, he has taught you the central dogma. And so
Yes, sir.
when a gene is expressed, what we can do is transcribed. The transcript is the gene product. The transcript is the gene product. That transcript, which will be RNA, we have different types of RNAs. We have, for your level, I think it's just 3 that you are supposed to concern yourself with, which is your tRNA, your rRNA, and your mRNA. Of these 3, mRNA is the one that is translated. So your gene products there can refer to the transcripts from your gene when it is expressed. Now, please note, your ribosomal RNA is produced in the nucleus, and then as per fibrosa when it is fiber-like. Once it is produced, it interacts with proteins, and then it's now called per granulosa. Okay. Then your tRNA is your transfer RNA, and that will be shipped into the cytoplasm. That is not produced in the nucleus. It is shipped into the cytoplasm, where it folds up to form a clove-leaf structure, and the function of that is to transport amino acids. Then you have the mRNA, which is the messenger RNA. Messenger RNA is one that has a code that needs to be decoded in the cytoplasm. And it is ribosome that will decode what is on the mRNA and assemble the corresponding proteins encoded by the mRNA. The corresponding amino acids will be linked together by peptide bonds to form a polypeptide, which will fold up to form the functional protein. Jemima, sorry. Williams
So where
Jemima.
is mRNA and tRNA made?
In the nucleus. You have a lot of genes, a lot of chromosomes. We have in humans, we have 23 pairs of chromosomes. Okay? In that nucleus, right? The region, that nucleus, what it does, what nucleus is there for, is to produce RNA, ribosomal RNA that makes up ribosomes, and also produce the proteins. It doesn't produce proteins. Proteins are brought into the nucleus from outside. Okay, those small proteins, they are brought into the nucleus from outside, and then they complex with the rRNA to form fibrous granulosa, and that is shipped outside the nucleus. When these components get outside, they are assembled together to form the large subunit of the ribosome and the small subunit of the ribosome. Those two parts of ribosomes will not come together until there is mRNA in the cytoplasm. Is it clear? Is it clear?
Okay. Yes, sir.
Yes, sir.
Yes,
sir.
Yes, sir. Yes, sir.
Can go on then. All right then. All right. Good. Then yes, this is showing us the heterochromatin close up and the euchromatin. I said heterochromatin is transcriptionally inactive. So it means here, Jessica.
Sorry, sir. Sorry to cut you short, sir. Sorry, please, can you go back? I was not hearing you. I was having network issues with the mRNA side.
That's not a problem of the teacher. Sir, please go on.
Wait, no, calm down. Just
calm down.
Okay, it's okay. It's okay. Just calm down. All right. Thank you.
Thank you, sir.
mRNA, tRNA. I told you that we have over 20 types of RNA. We have small heterogeneous, we have RNA, we have a lot of them. For this, your syllabus, you are concerned with only 3, which is ribosomal RNA, transfer RNA, and messenger RNA. Ribosomal RNA, transfer RNA, messenger RNA. Now, ribosomal RNA is produced, some of it is produced in the nucleus. In the nucleus. Transfer RNA. Remember, the nucleus is present inside the nucleus. Okay? The nucleus is present in the nucleus. The nucleus is not membrane bound. It's just a region within the nucleus that produces those RNA. Now, transfer RNA is also produced in the nucleus. Messenger RNA is produced in the nucleus. Once transfer RNA is produced, it is moved out of the nucleus through the nuclear pore complex, and when it gets to the cytoplasm, it folds up to form a clove-leaf structure. And the function of that in the cytoplasm is to transport amino acids about. Messenger RNA is single-stranded, and it is made from DNA that is within the nucleus. When a gene is expressed, the expressed product is called a transcript. Okay? Whatever RNA it is made is called a transcript. So the messenger RNA gets a code, is that a code on it. When it gets to the cytoplasm, ribosome will locate it and decode it. Ribosome will read the nucleotide in 3s, okay, and assemble the corresponding amino acids together. That means it joins amino acids together based on the code on the RNA, mRNA. And then when it has done, you have a polypeptide made, and that polypeptide will now fold up to form the secondary structure of the protein. The secondary structures will interact with each other to form the tertiary structure of that protein. Okay? Now, if a protein has subunits, then the different subunits come together to form the quaternary structure of that protein. Now, for the ribosomal RNA, when it is produced at first, it is called par fibrosa. Par fibrosa, because it's fiber-like under, when you look at it under the electron microscope. You can't see that with the light microscope. Then it is now complex with proteins, and it's now called par granulosa, because it looks like grains under the electron microscope. Is that clear enough?
Yes, sir. Thank you so much, sir.
Okay, so this is telling you, this is still an electron micrograph, but not so much detail. The NE is nuclear envelope. Par granulosa is PG. Par fibrosa is PF. And nuclear organizing region is NO. Okay? All right. So the nuclear envelope, like I said, has two membranes. That is two lipid bilayers. Okay? It enclosed in a flattened Joshua. Yes.
Sir, in that previous slide, there was a region named NAC. What's that region, sir?
Okay, that is N-acetyl.
Sir?
All right. That is before we finish, I'll remember. I can't remember the name of that region right now.
Okay, sir.
But what is important for you is the fibrosa, nuclear organizing region, and the nuclear envelope. So your nucleolus is all of this. Okay? All right. Okay. Yeah. I've described the nuclear envelope, and I've said it is the lumen is continuous with the rough endoplasmic reticulum. We know why rough is called rough endoplasmic reticulum, because it is studded with ribosomes, and it appears like that under the electron microscope. So, yeah, that's the space between the outer and inner membrane. It's also continuous with rough endoplasmic reticulum space. Yeah, the nuclear is enmeshed in a network of filaments. That's nuclear lamina for stability. So that means the nuclear lamina is just under the nuclear envelope, the inner membrane of the nuclear envelope. So the lamina consists of intermediate filaments, which are 3 to 10 nanometers thick. They are polymers of lamine. Okay, this lamine ranges from 60 to 75 kilodaltons. Kilodaltons is the unit we use for proteins, just like we use base pairs for nucleotides. We use kilodaltons to get the sizes of proteins. Okay. A-type lamina is inside, next to the nucleoplasm, while the B-type is near the nuclear envelope. That is inner membrane of the nuclear envelope. Yeah. And they may bind to integral proteins inside the membrane. The laminas are involved in functional organization of the nucleus, and they play a role in assembly and disassembly before and after mitosis. Okay, remember when you were taught mitosis, there is a stage where the nuclear envelope, they call it nuclear membrane for you, where the nuclear envelope disintegrates. And then the chromosomes align at the equator. So after the telophase, the nuclear envelope will reform around the two DNA that is in the soon-to-be daughter cells. So phosphorylation triggers disassembly. Phosphorylation of the lamina triggers disassembly of the lamina and causes nuclear envelope to break up into vesicles. Dephosphorylation reverses this and allows the nucleus to reform. So in the nuclear envelope, there are pores, and those pores will allow water, ions, and ATP to pass through. Other materials are regulated by the gatekeeper, which is the pores, like proteins in the pores. The nucleus is the site of ribosome assembly. I've already said that. The subunits of ribosome are assembled there, shipped outside, and then they are coupled together to form the large subunit and the small subunit. Mitochondria. Any questions on the nucleus? Any questions on the nucleus?
No, sir.
No, sir.
Right?
No,
Okay, so
sir.
No, sir.
mitochondria is plural for mitochondrion. Singular is mitochondrion, plural is mitochondria. So this organelle provides energy for cellular function. It provides energy for cellular function. Okay, and it's a double membrane bound organelle. That means it has two membranes. Just like the nucleus, it has two membranes. But here, the two membranes are not together. There's a space between the two membranes. The inner membrane is thrown into folds called cristae. The inner membrane is thrown into folds called cristae, on which you have your electron transport chain. Okay? So basically, molecules are broken down to obtain energy within the mitochondria because the mitochondria generates ATP. ATP is the energy currency of the cell. A cell needs ATP to be alive. A cell needs ATP to survive. A cell needs ATP to do work. Abayomi, yes?
Sir,
Ogule,
yes, sir.
yes?
You talked about two membranes and the inner membrane. I didn't get that part of the inner membrane, sir.
Okay. All right. No problem. So mitochondria is described as the powerhouse of the cell. Yeah, because it produces energy-rich molecules for the cell. And what are these energy-rich molecules? ATP. Mitochondrial genome is inherited maternally in several organisms. Okay, it is sausage-like organelle. Now, the double membrane divides its lumen means inside, into two distinct aqueous compartments. The double membrane divides the lumen into two distinct aqueous compartments. The inner compartment is called the matrix. That is, the inner membrane folds up into cristae, and the center of that one is called the matrix. The outer membrane forms a continuous boundary with the cytoplasm. And so there's a space between the inner membrane and the outer membrane. So they may vary in size and shape, but usually they are oval in shape. They are the site of aerobic respiration. And since they have their own circular DNA, note, this circular DNA resembles the DNA found in bacteria. Okay? The circular DNA resembles DNA found in bacteria. And some proteins found inside ribosome and inside the mitochondria also have been found in bacteria. Okay? Again, the ribosome found inside mitochondria is the 70S type of ribosome. Remember, we said ribosome, two types. You have the 70S, that's the intact ribosome 70S, and you have the intact ribosome 80S. The 70S is what is found in prokaryotes, and the 80S is what is found in eukaryotes. Here, what I'm saying is that inside the mitochondrial matrix, you have ribosomes. Those ribosomes are 70S ribosomes, and those ones help in protein synthesis. The structure of the mitochondrium is that it produces some of the protein it needs, while most of the protein it needs is coming from the genome, from the nucleus. The nucleus makes mRNA that directs proteins to be made for mitochondria. Questions? Any questions yet?
Yes, sir.
Questions?
Yes. Sir, I don't understand the meaning of nuclear lamina. lamina.
Nuclear lamina is the cytoskeleton. Is made up of intermediate filaments, and it's what keeps the shape of the nucleus.
Okay. Okay.
Yeah, but those intermediate filaments, their subunits are called lamine. There are two types.
Okay.
Yes, lamine. Those are the ones that are 50 to 60 kg kilodalton in size. Okay.
Yes, sir.
There
Okay.
are two types. There is the A-type lamine and there is the B-type lamine. The B-type lamine is the one you find close to the inner membrane of the nuclear envelope. Both of them are intertwined. The A-type is the one that is on the boundary of the nucleoplasm. Nucleoplasm is all the things that are inside the nucleus.
Yes.
Right?
Yes, sir.
Okay, so is that Aisha or Joshua?
Yes, I have a question, sir. That I didn't understand what you meant by I didn't understand the explanation you gave on the types of mitochondria, the outer and the inner stuff.
Okay. Then Joshua, what is your own question?
Sir,
Joshua Ishola.
Yes, sir. In the previous slide, you said that the mitochondria possesses RNA molecules. Those RNA molecules, are they also derived from the nucleus?
No. No, they are not derived from the nucleus. They are derived from the mitochondria itself because mitochondria has its own DNA, which is circular DNA, and it acts as ribosomes. And the ribosomes there, the type is 70S. So if it has a DNA, that means the DNA can be transcribed, okay, to form mRNA. And then since it has ribosomes, that ribosome can interpret code on the mRNA and make the corresponding polypeptides, which fold up to form the protein. What I added was that although mitochondria can make proteins for itself that will work inside it, it requires other proteins that are made by the nucleus, that are directed to be made by the nucleus. So it's like it makes its own protein and receives additional proteins from courtesy of the nucleus. Mitochondria is in the cytoplasm. Okay. Okay, so maybe now, okay, yeah. Aisha, your question.
Yes, sir. I said that. Could you explain that types of mitochondria again, that inner and outer stuff?
Okay, mitochondria is one of the double membrane organelle. Means it has two lipid bilayer. In the cell, it is one lipid bilayer that forms the cell membrane, plasma membrane. It's one lipid bilayer. Okay. In mitochondria, you have two lipid bilayers. Okay, forming the mitochondrium. So, and those two membranes are not joined together. They are not like the nuclear envelope that is pinched together. They are two separate membranes. One on the outside, then there's a space, and one on the inside. The one on the inside is thrown into folds, okay? And those folds, we call them cristae, okay? It's thrown into folds that we call cristae, all right? And because of these two membranes, the mitochondria has what we call a matrix, which is the innermost part, okay? Matrix. And then you have the other part, the space. Okay, so it's like compartmentalization. So you have two compartments in mitochondria. In mitochondrion, the space between the two membranes is one compartment. Then the inner membrane is what is inside the inner membrane is another compartment, and that is called the matrix. Is that clear?
Right, sir. Thank you, sir. Yes, sir.
All right. Okay, so now will be a good time. Let me introduce you to something called the endosymbiotic theory. Endosymbiotic theory. Okay. Okay, we just have enough time for it. All right. So the endosymbiotic theory is telling us simply that a long time ago, one primitive eukaryotic organism swallowed a prokaryotic organism, and they started living together symbiotically. Okay, then the prokaryotic organism now noticed that this eukaryotic organism is providing food for me, so I don't need my cell wall. So the prokaryotic organism lost the cell wall. Okay, and the prokaryotic organism said, okay, well, since this guy is producing food, let me provide energy for it. So is that prokaryotic organism that was swallowed that became the mitochondria? All right. So the scientist Lane, remember I mentioned the scientist called Lane, Miguel Lane. She's an American scientist. She is the one that proposed this theory. Okay, based on the fact that when they looked at the mitochondria, the ribosomes inside mitochondria, it was similar to the ribosomes found in bacteria. And when they looked at the DNA, the circular DNA found in mitochondria, it was similar to circular DNA found in bacteria. So she came up with that theory that one of the old eukaryotic organisms must have swallowed a prokaryotic cell. Okay, and so that organism that swallowed the mitochondria became the animal cell. Okay, now again, one animal that another cell, an old prehistoric eukaryotic organism swallowed another bacterium. Okay, and that bacterium lost the cell wall and was now this bacterium was a photosynthetic bacterium, so it could provide food for the eukaryotic organism. And so this eukaryotic organism now swallowed another bacterium, which was producing energy for it. Okay, that is now became the mitochondrion, and so you have the chloroplast and the mitochondrion, and that eukaryotic organism now became the plant cell. Okay, the one that did not swallow the photosynthetic bacterium is swallowed only the one that produces energy is called the animal cell. So animal cells don't have a cell wall. Plant cells have a cell wall. Okay, plants also have, in addition to mitochondria, they have the chloroplast. Okay, chloroplast. All right, this is one of the open chloroplasts. Yeah, this is the mitochondria. So plant cell, animal cell have a lot of things in common. What distinguishes them, what separates them, is the cell wall, chloroplast, okay, and maybe centriole. That reminds me, last class, somebody asked the question on why plants, lysosome, are not in plants. Okay, two reasons. The plant has a cell wall which screens off anything coming from outside. Second is that the vacuoles do the work of the lysozyme in plants. The vacuoles do the work of lysozyme. So lysozyme is majorly found in animal cells. Okay, let me leave room for questions. So that, yeah. Questions, please.
So,
yeah.
Could you lay that theory practical? Sorry, sir. That's
endosymbiotic theory.
Yes.
No,
yes.
I'll still talk about it. Simply, a long, very, very long time ago,
Okay.
a prehistoric, a primitive eukaryotic cell. There are so many answers. A primitive eukaryotic cell swallowed a bacterium. And that bacterium, they were living together symbiotically. Okay? Symbiosis means they had mutual benefits. That's this one was gaining something from this, and this one was gaining something from this. Over time of living together, the bacterium inside lost the cell wall and lost a lot of the organelles because it was protected and it was getting food.
Okay.
So the primitive eukaryotic that swallowed it was providing food for it because it can engulf things from the environment and digest it and give it to the bacterium. The bacterium said, okay, let me just be making energy for this guy. So that's how the animal cell evolved. The same way, a primitive eukaryotic organism swallowed two bacteria, okay? One photosynthetic bacterium and the other bacterium. So the photosynthetic bacterium lost the outer cell, the cell wall, and some of the organelles, and then started producing food from sunlight. Okay, the other bacterium lost the cell wall and most of its organelles and started generating energy for the bacterium and for the eukaryotic cell. And so they all started living together, and that one evolved to become the plant cell. So that's why, excuse me, that's why plant cells have both mitochondria and chloroplast. And that is why animal cells have mitochondria. But the mitochondrial the DNA in animal cell is circular. The DNA in chloroplast, the DNA in mitochondria is circular. The DNA in chloroplast is circular. The DNA in bacterial cell is circular. The ribosome found inside mitochondria is 70 years. The ribosome found inside chloroplast is 70 years. The ribosome found inside bacteria is 70 years. That was how these scientists deduced this endosymbiotic theory. So in eukaryotic cell, aside from the nucleus as the bulk of the genetic material as chromosomes.
Okay.
Now, mitochondria also has its own DNA and it can replicate on its own without the cell replicating. Okay? It can do things on its own because it has DNA that can control activities within it. Chloroplast same can replicate on its own. It can do things on its own because it has its own DNA. Both these organelles are within eukaryotic cell. Is it clear?
Yes, sir.
Okay. Oladipo Bisuga. You wanted to say something? Bisuga.
Good morning, sir.
Morning.
Yes, I want to get more insight on those what you talk about 70 years and 80 years.
Oga, stop wanting to ask your question. That when you say you want to do something, it means you are not ready to do it.
All right, sir. That's those 70 years and 80 years mitochondria. Can you
No, it's not 70 years and 80 years mitochondria. It's ribosomes.
Ribosomes, yeah, sorry.
Yes.
I'm
saying mitochondria has ribosomes inside it. And the ribosomes inside the mitochondria is different from the ribosome in the cytoplasm of the eukaryotic cell. In the cytoplasm of the eukaryotic cell, yes, the ribosome there is 80 years. And the one that is on the rough endoplasmic reticulum, the ribosome is 80 years. But inside the matrix of the mitochondrion, the ribosome there is 70 years. Chloroplast has circular DNA. Chloroplast has ribosomes inside it, okay, which is 70 S ribosome. In the cytoplasm of the plant cell, the ribosome there is 80 S. And the 70 S ribosome is similar to the ribosome found in eukaryotic organisms, found in bacteria. The circular DNA is similar to circular DNA found in bacteria. Is that clear?
Okay, so we have another class now.
Yes, so I have to stop.
We have another class. Okay,
so we have to have a class on Wednesday. We have to have the class on Wednesday. So I think
okay, no problem.
People say they're in systems engineering, right? So which means they can be available at 8. So maybe we'll make it 9 to 10.
Okay, thank you, sir.
Okay. All right then.
Yes, sir.
Have a lovely week ahead.
Okay. And by that time now.
Thanks, sir.
All right.
Okay,
but
thank you, sir.
if it won't work, you let me know on time so that one can take the necessary action. So we can do it 8 to 9 or 8 to 10, whichever one. So you let me know on time so that I can prepare for it. All right then.
Okay, sir.
Have a lovely